Hi everybody, welcome to this week's live stream. It is week 9. Today we'll be doing DNA replication, transcription, and translation. Alright, let's get started. Hope everybody is doing well and getting excited for the long weekend. Alright, an E. coli cell that contains a single circular chromosome made of double-stranded DNA is allowed to replicate for many generations in N15 medium. This is just due, it's an isotope, it just has to do with the weight of the atom. And hello, hope you are doing well too. I'm doing well, thank you for asking. Until all of the E. coli cells' DNA labeled with N15. So it's just in, we're starting with this isotope. One E. coli cell is removed from the N15 medium and is placed into a medium in which all of the available nucleotides are N14 labeled. The E. coli cell is allowed to replicate until 8 E. coli are formed. Given the situation, which of the following is true? To help us navigate this multiple choice, I drew a picture down here. First, I'm going to go through our answers and then check with the diagram I drew below. So A, some N15 labeled DNA will be found in all 8 cells. Some 15N labeled DNA will be found in only 4 of the cells. Some 15N labeled will be found in only 2 of the cells. And when they're saying 4 of the cells, they're saying 4 of the 8 produced, as we see here, or 2 of the 8 produced. Some of the 15 unlabeled DNA will be found in only 1 of 8 of the cell. So if we look at the diagram that I drew below, right here we're starting with 2 N15. So a line straight through is N15. A dotted is representative of N14. So once they replicate, and they just remember that this has been placed in N14 solution. Once it replicates, it's going to use that N14 DNA to replicate with, thus producing, we're kind of splitting these two down the middle, replicating with this N14. But once we replicate again, we're still in that N14 solution. And so this actually needs to be changed. I try to draw these things ahead so I could get through them with you guys more concisely. So in that case, we're going to be then splitting down the center of these 2 lines. And we're still in that N14 medium, so we're going to be producing N14 here, strands, N14 strands, and then those strands that were originally N15 in the very, very beginning. So at the very end here, we're seeing that same replication pattern with 8, and we still only have 2 of those strands that are N15 from the original because of the solution they were in, making C the correct answer. Great. Okay, moving on to 2. This will be seen on the next page, but I'm just going to go through the systems with you before we get started. So 2, draw a model of a short segment of double-stranded DNA. Your model should describe the roles and relationships of all the following enzymes and structures in replication. So parental DNA is a parental DNA that you start with. It's going to have 2 strands, and that's what you're going to replicate. Anti-parallel strands are just the segment in which the DNA is placed when double-stranded. both of them go to the 5' to 3' direction, but because they are anti-parallel, they are complements of each other. So 5 and 3 will go together on both ends. Single-stranded DNA is just one of these guys. Binding proteins on the leading strand help with the binding of polymerase. And the replication fork is where replication begins. Where replication begins. And I just wanted to double check my definition on single-stranded binding proteins. So they bind to the single strand of DNA during replication that is newly separated, so that keeps it separated. So I'm so sorry. It doesn't aid in polymerase binding. What it does is when the DNA strands are separated, it just kind of puts a lock and key on that to make sure that it doesn't move. Replication bubble is the segment also which DNA is occurring. So if DNA normally looks like this and it's replicating, we can see it kind of do this so that we're getting new strands in and out. So this thing is the replication bubble. Looks kind of like a bubble. The lagging strand is the strand of DNA replication that is in opposite direction. DNA likes to replicate in the 5' to 3' direction, so when you're separating the DNA fragment, there's going to be one of those ends that ends in a 5' direction, thus going from the 3' to 5' direction leaves these fragments because they can still only be synthesized in the 5' to 3'. I know that was a lot of words, but I drew a picture to help. So 5', 3' end we've talked about. RNA primers are what help anneal it makes the first few sets of DNA so that it can just start going. You can't replicate out of nowhere. You have to start somewhere, and that's where the primer comes in. Origin of replication is where it begins. Okazaki fragments are a result of the lagging strand. It's those little fragments of DNA that are made a little at a time. DNA polymerase is what synthesizes polymerase. There's 1 and 3. Helicase is what unwinds the DNA. DNA ligase joins Okazaki fragments. And primase is what helps with RNA primers annealing. So in this diagram I have drawn on the next page, I'm just going to go ahead and label those things that we talked about. In this case, this would be the 5', 3', therefore 3', 5'. This right here is the parental DNA. This right here is helicase, which helps unwind the DNA. And these two guys right here are the single-stranded binding proteins that are helping keep these DNA fragments separated from each other. This red thing right here is that RNA primer that we talked about that helps replication get going. This enzyme attached is primase, which, as you can see from the diagram, is kind of creating that primer that's annealing to the free DNA strand, that single DNA strand. This molecule right here is DNA polymerase. This is a version of it. Polymerase 1. Don't get lost in the weeds. It's just for the diagram. This is another polymerase. As you can see, it's synthesizing. If we said that this was 3'. It's synthesizing in the 5' to 3' direction. And this DNA polymerase is also synthesizing in the 5' to 3' direction. And now that you can see more clearly why we get those Okazaki fragments. I said this was DNA. This strand that can be synthesized continuously is called the leading strand. This strand with the Okazaki fragments that come in these segments is called the lagging strand. And this blue enzyme here that is helping anneal these two fragments together is called DNA ligase. DNA ligase is responsible for just joining those two fragments together. And this guy, I'm just going to label this fragment as an Okazaki. Okazaki. Oh my gosh. My apologies to Dr. Okazaki who found these fragments because I've been butchering his name and it took me forever to spell it. But anyway, that's what those fragments are. Right here, where we see the DNA splitting, is called the replication fork. And then, like I showed earlier, this is what would be called the replication bubble. And that's basically this whole segment. But that looks really messy, so I'm just going to do that. Okay. So I hope that this was helpful. I kind of ran through that. Are there any questions? Okay. The replication bubble is just where the replication is happening. So, DNA, when it's together like this, would it look like that, but when it's being replicated on and it separates like this, if this were to be drawn like this, you could see them coming together again, and that's why it just looks like a bubble. So that's why they call it the replication bubble. Yeah, so that's where the whole process is. That's where replication is happening. So it's important to remember that only certain parts of DNA get replicated to go into proteins. Some of our segments of DNA aren't used, so there's just specific segments of it. That's why you get these bubbles in the DNA for replication, because we only need a specific part of it. They don't both go towards the replication fork, and both of the lagging strand and the leading strand are utilized. I'll draw something down here. So let's say this DNA is getting replicated, We'll make the top the leading strand, the bottom the lagging strand. So both of these DNA strands that are now single are both going to get replicated. Neither goes away. So this oh shoot, I always do this. This blue would be, if this is what's being replicated, the blue would be going, synthesizing in the 5' to 3' direction, which makes this the leading. And because the lagging strand still has to be synthesized 5' to 3', it can only go into segments. So if it starts, both of them start at the replication fork. So let's say that the replication fork, the fork is here. It can still synthesize from the fork. So it's going to start somewhere here and go and be like, oh, I gotta stop. And then when that DNA keeps getting unwound, then it's going to make another one. And yeah, that's different from introns and exons. Those are, that's kind of next step. So similarly to this, we saw how these two got replicated into these four. That's still what's happening here. Like, but both sides will get replicated. It's just that one side can be done all the way through because it's going from 5' to 3', and this is unwinding that way. So as it's unwinding, it can keep going down that way, 5' to 3', just continuous. But because it's unwinding that way, and this is the opposite direction, it'll start here and try to keep going, but then it'll have to stop because it's not completely unwound yet. And so I hope that makes more sense. And introns and exons, are the next step. So once the DNA was replicated or it's made into mRNA, the mRNA then is what deals with introns, exons, what do we need to get rid of, what is actually useful. And you'll actually see that in a couple of questions, so I'll be able to explain that a little better. And to answer the next question, the replication bubble, yeah, is just inside some part of the DNA. So this is the DNA itself, and therefore it's just when the DNA is separated, that little section is what the replication, oh my gosh, the replication bubble is. So I hope that cleared things up. Okay, number 3. Some researchers estimate that the mutation rate for any given gene or its DNA in certain strains of bacteria is about 10 to the negative 8. This means that one error or mutation in a given gene is introduced for every 100 million cell divisions. What can cause mistakes in replication? So mismatched bases can cause mistakes. Anytime during replication, DNA polymerase is not perfect, so sometimes you can match the wrong base pair together, but also alternatively, things like UV, X-ray, radiation, or just other chemicals that modify the DNA so that errors are perpetuated during subsequent replication events could cause mistakes in replication. So we're told to protect our eyes with sunglasses, protect our skin with sunscreen. That's because the UV coming from the sun can then damage our cells. And I am not sure the answer to your question. Let me look in my notes really fast. So what I'm finding for things like UV damage, because mutations can obviously be inherited, but in terms of like UV damage or things that you're accumulating on external, the only thing that's inherited are things like hotspot genes. So hotspot genes are places where mutations are really likely to occur, or just that mutations occur a lot there. So if you have this part in your DNA that's a hotspot gene that's really sensitive to UV light and can cause things like melanoma, then that can be genetically linked, but not the UV light itself, because you can avoid things like sunlight and X-rays. One second. Of course. How are such mistakes normally corrected? Normally, DNA polymerase inserts a new nucleotide during replication, and while it's doing that, it also checks and proofreads each nucleotide against the template for any mistakes or mismatches. So if a mismatch is found, DNA polymerase has the ability to remove and replace it with correct nucleotide. But errors introduced by X-rays and other factors are generally repaired by different DNA repair enzymes that are specified for that kind of function. So, corrected by DNA polymerase's ability to proofread. Okay, awesome. So this is draw a model for intron removal, which contains pre-mRNA exons, RNA splicing, spliceosome introns. So here's something that'll probably help answer your question that you had earlier. So this is what we're, these small RNAs are what is also drawn over here. This is 5' end, this is 3' end, and these RNAs are what just came off this DNA. So if we're trying to, we'll tackle, I know this is getting muddy, sorry about that, but if we're trying to replicate that part of the DNA, let's say we just got our mRNA from there. It's going to still have its introns and exons. I kept saying ectrons, oh my gosh. Introns and exons. It hasn't yet been spliced of those. So what those encounter is just that the exons are what carry the information that we need. Sometimes gene sequences are not just all together perfectly for us, and important segments are separated by bases that don't really have anything to code for. So we're just taking those things out so that when we're translating it and we're reading it, we're just cutting the fat out of it. It makes sense. Instead of saying like, I put on my shoes to go to the park that was 1.5 miles away for 30 minutes today. Your mom doesn't, care. She just wants to know, like, hey, I went to the park today for a few. Okay, great. Your mom cares. Shouldn't have said your mom. Anyway, I'm so sorry. So this here is a pre-mRNA. This thing, forgot to switch back to pen. That's highlighted a darker orange is an exon. We'll call this exon 1. So this guy over here is exon 2. This right here is the intron. And so once it's spliced out, this is the cut out intron. And these two exons are joined together here. This is exon 1 and exon 2. 5', 3'. And then this is what our mRNA is. And that encodes the genetic information needed to synthesize proteins. Okay. And just so you guys know, this worksheet today is pretty long. I may not be able to finish it all by 6, but seeing the holiday and that, finals are coming up in a couple weeks, I'm sure you guys have things to study for. I'm still going to cut it off at 6, and the key will be released to you with all of the answers and for you to see those yourself, but I'll definitely try to get through as much as possible. Just wanted to give you guys a heads up. I'm also going to put attendance in the chat. Okay. So, if 20% of the DNA in a guinea pig cell is adenine, what percentage is cytosine? Explain your answer. We know A and T go together and C and G go together. So for every A, there is a T. So we can also say that A is equal to T, C is equal to G. If A equals 20%, T is equal to 20% as well. And if we're doing this out of 100%, then A and T together account for 40% of the total 100. So therefore, we know that C and G and C, or C and G, are equal to 60%, because 100% minus 40 is equal to 60. Therefore, this divided by 2 is what C and G both account for. Therefore, cytosine is equal to 30%. Okay. A number of different types of RNA exist in prokaryotic and eukaryotic cells. List the 3 main types of RNA involved in transcription and translation, and answer the questions to complete the chart. The first type of RNA we're going to talk about is mRNA. And it is produced in the nucleus from structural genes on DNA. And structural genes just mean specific genes, but that's what they're usually called. The mRNA functions in the cytoplasm, and it's translated into protein because it carries the information in codons that determine the order of amino acids. And AA is just what I write for an abbreviation for amino acids. The next RNA is tRNA. And it's produced also in the nucleus by other genes that code for tRNA. And then where and how does it function? It functions also in the cytoplasm. And tRNA combines with a specific amino acid through complementary base pairing of tRNA with the codon, and they have A, P, and E sites, and so the ribosome brings the correct amino acid into the position, and it grows the polypeptide chain. So tRNA is responsible for creating the polypeptide chain. So tRNA, just to give you guys a visual, kind of looks like, it's like a T shape, but I'm going to draw it as like, I think this makes it look easier. So these three sites are what's responsible with binding with a complementary codon. So these bind with like C, A, G, or something like that, and it's carrying an amino acid. And so this binds and attaches to another one, and then we'll have like another tRNA that'll come here and sit next to this one and then attach to this one. And then the chain just kind of grows and grows like that. And yeah, AA is an amino acid. The next RNA is our RNA. This is our final RNA, and R is ribosomal. So this is the same place. It's nucleus, structural genes on DNA. And this is also in the cytoplasm. All of the mRNA all the RNA are found in the cytoplasm. That's where all this kind of goes down. And our RNA molecules combine with a protein to form the ribosomes that serve as the base of interactions between mRNA codons and tRNA anticodons in the translation in the cytoplasm. Oh yes, totally can. Sorry about that. Yeah, no problem. Okay, sorry. Where was I? Combines with protein to form ribosomes. Alrighty. Cool. Alright, number 7. It's just having us go through and translate this. These are kind of fun. So if it's the non-template strand, the template strand would be complementary to this. So we start with 3 prime, T, A, C, A, T, A, C, G, G, T, T, A, C, G, T. And remember it says DNA, so we're not substituting T for U. And then mRNA would then be complementary to this template strand, but would substitute U for T. AUG, UAU, GCC, AAU, GCU. Alright, and then the anticodons. We're splitting this up into 3s. I'm just going to highlight every other one. So the anticodon, complementary to this guy, would be U, A, C. And remember, this is tRNA, so we're still using U. A, U, A, C, G, G, U, U, A, C, G, A. Oh, I realized I got something wrong. This is actually supposed to be A, and this is supposed to be U. Okay. So we're going to use this template strand to answer questions, and I'm going to copy it down below so that we can see it. So it says that this DNA strand produces mRNA. What does the sequence of the mRNA read from 5' to 3? Our sequence is starting with 3', TAC, GCA, AGC, AAT, ACC, GAC, and GAA. So then the mRNA from 5' would just be complementary to that. Alright. And then it's asking for what sequences of amino acids does this mRNA code. And we just use this chart up here. So we start with this one, A, and then we go to this column. So this is 2, this is 1, and then these little ones in here are 3. So you start with A, U, G, A, here, U, and then G. So then we get methionine, for this one, CGU, CGU, we get arginine, UCG, UCG, we get serine, I'm going to put this down here, UCG, and then UUA is leucine, UGG is tryptophan, and then CUG is here, CUU is here, and both are leucine. Okay. Okay. The chart lists 5 point mutations that may occur in the original strand of DNA. What happens to the amino acid sequence of protein produced as a result of each mutation? Note: position 1 refers to the first base at 3' end of the transcribed strand. The last base in the DNA strand at the 5' end is at position 21. So I'm just going to go ahead and number these. Okay, now I know those are small, but it'll just help me know what positions are where. Sorry, I thought I had to sneeze, but it went away. Anyway, so substitution of T for G at position 8. Position 8 is right there. This little highlighted G. And a substitution of T for G, we have AGC. This would change this to ATC. So the codon then, if it were to replace this, would be UAG. And so what this would do is, because UAG is a stop codon, translation would stop at this point and would create a short polypeptide. And this short of a polypeptide would likely be non-functional. So transcription is not stopping, translation is in fact stopping. Transcription is when you're moving from DNA to mRNA, and what this is specifically talking about is I'm talking about something like almost 3 steps further, where we've already translated to mRNA, and now we need to code for tRNA. And so if this is creating this codon on the mRNA, then translation to proteins is not going to occur because it is now coding for a stop codon. So a UAG in the mRNA is not going to stop it from replicating. Changing this in the DNA is not going to stop replication from happening, but when you get to the translation into protein, the codon will code to stop. Okay, awesome. Yeah, of course. And no, the last live review, I actually do have one week 10. It is also going to be on Tuesday before finals week. So next week there is one on Tuesday, and then finals week there is not one. A short. This is supposed to be likely non-functional polypeptide, though I know my grammar is off by putting the parentheses after it. Okay, addition of T between positions 8 and 9. We have AGC again, this is 7, 8, 9. This means that this would turn into AGTC. And what this would do is these codons would remain functional, the first two that can be and yeah, for sure I can totally do that. Just so you guys know, I did have Dr. Reddy last year, so I'm better versed on how that class is structured, so a lot of my pointers will be coming from that class specifically, but I can for sure just give you guys tips on how I study, and maybe it can help you guys. So what an addition of T would do is it's changing what used to be these 3 codons into now this codon, and then this C is going on and it's kind of bumping over those next bases. Like, I'll just complete some of these. So it's bumping over these bases. So now this is a codon, this is a codon. It's kind of moving and shifting it over. So serine would still be the third amino acid, but all amino acids that follow would differ from the sequence that was originally coded in this equation above. Okay, deletion of C at position 15. So deletion does something similar, though of course it is different than an addition, to an addition. So in this case, a deletion at position 15, which is this guy up here, would make the first 4 amino acids stay the same. But the rest be different. And slightly shorter because we wouldn't be using this last base to create an amino acid. Substitution of T for C at position 18. So we have 1, 2, 3, 4, 5, 6, 7. At position 18, we originally have GAC. Now this would be GAT. And if we reference the chart, then leucine is still inserted as a 6th amino acid. So nothing really changes. This is what we call a silent mutation. Awesome. Deletion of C at position 18. Also, no problem. Thank you for coming. So the deletion of C, now G, A, C, G is now going to be G, A, G. So deleting this guy still produces leucine, sorry. But we're only given part of the sequence, so we also don't know what the next part of the chain would be because it's near the end. Okay, and then which of these mutations produces the greatest change in the amino acid sequence of the polypeptide coded for by this 21 base pair gene. So it's just asking, out of all these options, which one does the most damage. And yes, you're right. On that last one, it would get a little shorter because we are deleting one. But all of this is saying with the answer is that from up here, because we're talking about this position, it's just saying we don't really know what the amino acid sequence would be because we don't know the rest of it. But yeah, you're right. It does delete it, and it makes the length shorter of the DNA. So the answer to this would actually be number 1, because the stop codon, it makes it so short that it's non-functional. And just remember, we have 3 stop codons. I like to remember these by you are away, you are gone, you go away, or more intuitively, these two would be switched. You are away, or you are annoying, you go away, you are gone. So because this produces stop codon early, the mutation produces the greatest change. So mutation of I, which is this one, produces the greatest change. Okay, and we are nearing the end here. I'm going to skim over the next couple just to show you guys what to look out for in the key. The next couple of questions have to do with the differences of amino acids, what it could do. So this one really quickly is just talking about a single base substitution that creates, that changes glutamic acid into valine. Glutamic acid is a part of hemoglobin molecule, which is what binds to oxygen and flows through the blood. So glutamic acid, as you can see here, this is its side chain highlighted in yellow, and valine side chain is also highlighted in yellow. The side chain on glutamic acid is actually polar, whereas valine is nonpolar. And so if you were to change that simple base substitution, changing it to valine would actually change its polarability within the hemoglobin molecule, making a structural change in hemoglobin, because valine would like to more so be within the protein rather than facing the exterior aqueous environment. So this could change the hemoglobin structure. I just found this cool, so I really wanted to go through this one really fast. Anyway, so yeah, the next one's going to just ask about X-ray and external mutation. The next true or false questions deal with the knowledge of replication and what enzymes do what. And then the last couple questions have to do with our ability to see what bands are complementary and what is RNA from DNA. Hint, hint, RNA always includes U. But yeah, that's the last couple. I'm sorry that we couldn't go over them. This DNA replication is always a lot of information. So of course, if you have any questions, feel free to reach out. So regarding a question given earlier about tips on timing yourselves for the exam, if you're in person or even not in person, for myself, I always like to wear a watch. I know that sounds like ancient, but I actually got that advice a long time ago. It's always helped, just to keep your eye on the clock and see how much time. And yes, the answers are written in the key. And just see how much time is left. I'm not sure how many questions you guys have on your final. From what I remember, I think we had like 80 questions in 90 minutes in Dr. Reddy's class. So what I would do for each midterm was set up a document with important figures and really study that. And I rewatched the lectures and I had handwritten notes of just important things that were said by the professor and things that are given of more importance. And so a lot of the figures is just going over it yourself, maybe make some flashcards. But the main thing that I would say for timing yourself for the exam is that don't, if you find a question more difficult than the other ones, you want to spend more time, I would say skip it. Try to knock out the ones that you feel most comfortable with first, and you can come back and spend time on those last few. What else? Process of deduction is really, really helpful. Remember to read every answer, go through it and be like, is this possible, is it not possible? I get really bad test anxiety, so I always like to breathe beforehand and like massage my temples, do all that stuff. Is there anything else more specific that you wanted advice on? Also, thank you all. It is 6, so of course if you have to go, no problem. I might stick around for a minute just to answer more questions. Awesome. I know, it's definitely very overwhelming, but don't get too lost in the weeds of it. I really do suggest just to take a breath, take a breather. You know a lot more than you think you do, I promise, because I always get lost in my head. I'm the type of person right before an exam, all of the information shoots out of my brain. I'm like, whoa, I actually have never looked at a biology book in my life. It's not true. You guys are all really good students, and I'm sure you're all studying very, very hard. So a trick that I like to do is a sequence of massaging, and it might look crazy, but it really does help me. So before tests, I'll close my eyes, and I'll start deep breathing, and I massage my temples for 7 rounds of circles. I massage my collarbones for 7 rounds of circles, and I massage my earlobes for 7 rounds of circles, and I just focus on that and do deep breathing. And then I smile because it relaxes your frontal lobe. So just remember to smile and breathe. You guys are going to nail it, I promise. And I'll be here next week if you guys have any more questions. But anyway, have a really nice long weekend. I hope you guys enjoy the holiday. And I hope to see you guys next week.